I'm here to discuss the health, ecological, and ethical concerns in animal biotechnology. So, what is animal biotechnology? Animal biotechnology is the use of science and engineering to modify living organisms. The goal is to make products to improve animals and to develop microorganisms for specific agricultural uses. Examples of animal biotechnology include creating transgenic animals, animals with one or more genes introduced by human intervention, using gene knockout technology to make animals with a specific inactivated gene and producing nearly identical animals by somatic cell, nuclear transfer or cloning. So animals that were used in biotechnology, animal biotechnology are called transgenic animals. So what is transgenic? Transgenics, also known as recombinant DNA, is the transferal of a specific gene from one organism to another. Gene splicing is used to introduce one or more genes of an organism into a second organism. A transgenic animal is created once the second organism incorporates the new DNA into its own genetic material. In gene splicing, DNA cannot be transferred directly from its original organism, the donor, to the recipient organism or the host. Instead, the donor DNA must be cut and pasted or recombined into a compatible fragment of DNA from a vector, an organism that can carry the donor DNA into the host. The host organism often is a rapidly multiplying microorganism such as a harmless bacterium, which serves as a factory where the recombined DNA can be duplicated in large quantities. The subsequently produced protein then can be removed from the host and used as a genetically engineered product in humans, other animals, plants, bacteria, or viruses. The donor DNA can be introduced directly into an organism by techniques such as injection through the cell walls of plants, or into the fertilized egg of an animal. This transferring of genes alters the characteristics of the organism by changing its protein makeup. Proteins, including enzymes and hormones, perform many virtual functions in organisms. Individual genes direct an animal's characteristic through the production of proteins. So in animal biotechnology, scientists insert gene to an animal to make them become genetic modified organism where, where the foreign gene will express itself. For simple definition or shorter definition, uh, um, a transgenic animal is an animal that express a foreign gene. And second is animal whose genome has been altered by the transfer of a gene or genes from another species or breed. There are different types of transgenic animals, such as transgenic rats, rabbits, pigs, sheep, cows, and fish. And 95% of transgenic animals are mice. So, as we see, mice are usually used in animal biotechnology, maybe because they are less in legal issues or ethical issues in manipulating of mice compared with other animals. So, before I proceed to the main topic, which is the health, ecological, and ethical issue, ethical issues in animal biotechnology. Let me first discuss the related issues, which is the potential benefits of animal biotechnology. The potential benefits of animal biotechnology are numerous and include enhanced nutritional content of food for human consumption, a more abundant, cheaper, and varied food supply, agricultural land use savings, a decrease in the number of animals needed for the food supply, improved health of animals and humans, development of new low-cost disease treatments of humans, and increased understanding of human disease. So let's proceed to the main topic. According to the survey, according to the survey conducted by Pew Initiative on Food and Biotechnology, 58% of those polled said they oppose scientific research on the genetic engineering of animals. And in a Gallup poll conducted in May 2004, 64% of Americans polled said they thought it was morally wrong to clone animals. So, what are the health issues or concerns? Concerns surrounding the use of animal biotechnology include the unknown potential health effects to humans from food products created by a transgenic or cloned animals, the potential effects on the environment, and the effects on animal welfare. So, before animal biotechnology will be used widely by animal agriculture product systems, additional research will be needed to determine if the benefits of animal biotechnology outweigh these potential risks. Next is ethical issues or animal experimentation. Animal experiments are widely used to develop new medicines and to test the safety of other products. Many of these experiments cause pain to the animals involved or reduce their quality of life in other ways. It is morally wrong to cause animals to suffer, then experimenting on animals produces serious moral problems. Animal experimenters are very aware of the ethical problem and acknowledge that experiments should be made as humane as possible. They also agree that it's wrong to use animals if alternative testing methods would produce equally valid results. So there are two positions on animal experiments. One is the position on animal experiment is in favor of animal experiments and against animal experiments. In favor of animal experiments, experimenting on animals is acceptable if suffering is minimized in all experiments and human benefits are gained with which could not be obtained by using other methods. And in against animal experiments, experimenting on animals is always unacceptable because it causes suffering to animals. The benefits to human beings are not proven. Any benefits to human beings that animal testing does provide could be produced in other ways. The last is the ecological issue of transgenic animals or the ecological concern to this genetical modified organism when released to the wild. where we don't know what could be the effect on the ecosystem simply because they are genetically modified. That's all. Thank you.